Dear friends, hearty welcome. The topic for our today's lecture is genetic code and characteristic features of the genetic code. Genetic code. Along we have already studied that the DNA is the master molecule present in the cells of living organism which regulates, controls, initiates and guides the entire process of protein synthesis. The DNA is the master molecule which initiates as well as it guides, it directs and regulates, controls the entire process of protein synthesis. आपण सगळेजण आधी शिकलेलो आहोत की डीएनए वर काही विशिष्ट माहिती असते, किंबहुना डीएनए हा प्रोटीन सिंथेसिस या प्रक्रियेवर संपूर्णतः नियंत्रण ठेवत असतो. ती प्रक्रिया कशी घडावी याला डायरेक्ट करत असतो, गाइड करत असतो. and that's why I said it is supposed to be the master molecule. Now the process of protein synthesis as regulated, guided by the DNA, therefore it must have certain necessary information, it must carry requisite information for the process of protein synthesis. याचा अर्थ डीएनए वर प्रोटीन सिंथेसिस योग्य पद्धतीने घडून येण्यासाठीची काहीतरी माहिती ही निश्चित असली पाहिजे. Now the basic genetic information present on the DNA, the basic genetic information present on the DNA necessary for the process of protein synthesis is present in the form of nucleotide sequence was first of all evidenced by two scientists, namely Yanofsky and Sarabhai. या दोन शास्त्रज्ञांनी यानॉफ्स्की आणि साराभाई यांनी सर्वप्रथम असं सांगितलं की प्रोटीन सिंथेसिस साठी आवश्यक असणारी बेसिक जेनेटिक इन्फॉर्मेशन हे डीएनए चे स्ट्रँड्स तयार करणाऱ्या न्यूक्लियोटाइड सिक्वेन्सच्या स्वरूपात किंवा नायट्रोजन बेसेसच्या सिक्वेन्सच्या स्वरूपात असतात. द फ्रान्सिस क्रिक फर्स्ट ऑफ ऑल सेड दॅट the basic genetic information necessary for the process of protein synthesis is present on DNA in the form of code language. Francis Crick first of all suggested that the basic genetic information present on DNA for the process of protein synthesis is present in the form of code language, and he called it as genetic code or maybe known as cryptogram. So the meaning of genetic code is very simple. Genetic code is nothing but it is the basic genetic information present on DNA in the form of code language for the process of protein synthesis known as the genetic code. And we know that this basic genetic information present on DNA is inscribed, is transferred, is printed on messenger RNA at the time of process of transcription. Thus the messenger RNA receives that genetic information in the form of some code words, and that code words may be known as codons. अर्थात ज्या वेळेला ट्रान्सक्रिप्शन घडून येतं त्यावेळेला डीएनए वर असणारी ही जी कोड लँग्वेज मधली जेनेटिक इन्फॉर्मेशन आहे, ज्याला आपण जेनेटिक कोड म्हणतो, ही मेसेंजर आरएनए वर छापून दिली जाते, इन्स्क्राइब केली जाते, प्रिंट केली जाते आणि मेसेंजर आरएनए वर ती इन्फॉर्मेशन काही जणू कोड वर्ड्सच्या स्वरूपात असते. कोड वर्ड्स याचा अर्थ सांकेतिक जणू शब्दांच्या स्वरूपात असते, ज्यांना कोडॉन्स असं म्हणतात. We are going to talk in detail about the codons and its nature after some time. The codons of messenger RNA are triplet in nature. Listen, the codons on messenger RNA are triplet in nature. That is, the codon is made up of 3 successive nucleotides. Codon consists of group of 3 successive nucleotides or 3 successive nitrogen bases was first of all suggested by scientist George Gamow. याचा अर्थ मेसेंजर आरएनए वर जे कोडॉन्स आहेत, कोडवर्ड्स आहेत, ते 3 न्यूक्लियोटाइड्सच्या ग्रुपने तयार झालेले असतात. Each codon consists of group of 3 successive nucleotides. It was first of all suggested by scientist George Gamow, and experimentally it was proved by Francis Crick that the codon is triplet of nucleotides. The nature of codon as triplet of nucleotides. याचा अर्थ प्रत्येक कोडॉन हा 3 न्यूक्लियोटाइड्सने तयार झालेला असतो. We shall talk again in very detail. So, the Francis Crick, the great scientist, first of all defined what is genetic code. Also, he suggested the meaning of codon, and also this term genetic code was proposed by the Francis Crick. So, after all, what is genetic code? Genetic code is nothing but it is the basic genetic information present on DNA in the form of code language. Okay, and I said that this basic genetic information present on DNA in the form of code language is transferred on messenger RNA in the form of code words. I told you that code words may be known as the codons. Just consider here is a single stranded messenger RNA with 5' end, 3' end, and these all dots, they represent single nucleotide, single nitrogen base. So the triplet of nucleotides, group of 3 successive nucleotides on messenger RNA are known as codons. It means that the basic genetic information, that is genetic code, is transferred on messenger RNA, and messenger RNA possess that information in the form of codons, and it was first of all explained and experimentally proved by Francis Crick. The actual nature of codon and genetic code later on deciphered, cracked, that is in detail studied and explained by 3 different scientists such as Marshall Nirenberg, Mathai and Dr. Hargobind Singh Khurana. या 3 शास्त्रज्ञांनी नंतर जेनेटिक कोड आणि कोडॉनचं स्वरूप, नेचर हे अतिशय विस्तृतपणे अभ्यासलं आणि एक्सप्लेन केलेलं आहे. I said that Marshall Nirenberg, Mathai and Dr. Hargobind Singh Khurana, they deciphered, they cracked, they studied in very detail the nature of genetic code by using artificial mRNA template in cell-free system. Artificial mRNA template in cell-free system by performing the process of protein synthesis. याचा अर्थ या 3 शास्त्रज्ञांनी कृत्रिम रीतीने मेसेंजर आरएनए तयार केला आणि सेल मध्ये नाही, सेल व्यतिरिक्त in laboratory they studied the process of protein synthesis to understand the nature of genetic code. Now we people have to understand why the codons are always triplet in nature. I said just now, codon is nothing but the triplet of nucleotides, group of nucleotides on messenger RNA. Now question arises, why are codons triplet नेचर. कोडॉन्स हे ट्रिपलेट का आहे? व्हाय कोडॉन्स आर नॉट सिंगलेट और डबलेट? So let me explain you why each codon consists of 3 nitrogen bases or 3 nucleotides. Means here we have to understand why each codon triplet in nature. Why each codon is made up of group of 3 nucleotides, group of 3 successive nitrogen bases. Let us consider. We know that there are presence of 20 amino acids which take part in the process of protein synthesis. आपल्याला माहित आहे की सेल मध्ये प्रोटीन सिंथेसिस या प्रक्रियेमध्ये 20 अमाइनो एसिड्स सहभागी होत असतात. And again you must know that each codon specifies single amino acid. Each codon specifies single amino acid. It means single codon carries information for only one amino acid. एक कोडॉन एकाच अमायनो ऍसिडला स्पेसिफाय करतो. याचा अर्थ एक कोडॉन एकाच अमायनो ऍसिडची माहिती ही कॅरी करत असतो. नाउ द मेसेंजर RNA consists of 4 different types of nitrogen bases, isn't it? Such as the adenine, uracil, guanine, cytosine. So I said that 20 different types of amino acid take part in the process of protein synthesis and each codon specifies single amino acid. आता जसं मी म्हटलं की प्रोटीन सिंथेसिस या प्रक्रियेमध्ये 20 अमायनो ऍसिड्स सहभागी होऊ शकतात आणि एका अमायनो ऍसिडची माहिती एकच कोडॉन कॅरी करतो. One codon specifies single amino acid. So obviously to specify 20 different types of amino acids, at least 20 different types of codons are necessary. Right? याचा अर्थ कमीत कमी 20 अमायनो ऍसिडला स्पेसिफाय करण्यासाठी 20 वेगवेगळ्या प्रकारचे कोडॉन्स हे आवश्यक आहेत. नाउ वी नो दॅट इन RNA there are presence of 4 different types of nitrogen bases, what here I have written. If each codon consists of only one nitrogen base, consists of only one nitrogen base or nucleotide, then how many codons are possible? समजा एक कोडॉन फक्त एकाच नायट्रोजन बेसने जर तयार होत असेल तर जास्तीत जास्त किती कोडॉन शक्य आहे? व्हेरी सिंपल. As there are 4 types of nitrogen bases and if each codon is with single nitrogen base, then maximum 4 codons are possible. Okay, such as the adenine containing, guanine containing, cytosine containing and uracil containing. And these 4 codons are not enough to code to specify the 20 amino acid. आणि अर्थातच हे 4 प्रकारचे कोडॉन्स 20 वेगवेगळ्या प्रकारच्या अमायनो ऍसिड्सला स्पेसिफाय करण्यासाठी निश्चितच पुरेसे नाही. नाउ कन्सिडर द सेकंड कंडिशन. सपोज इफ each codon is with 2 nitrogen bases or 2 nucleotides. समजा असं कल्पना केली की प्रत्येक कोडॉन हा 2 नायट्रोजन बेसेसने तयार झालेला आहे. इट मीन्स द कोडॉन्स आर सपोज टू बी डबलेट. तर हाऊ मेनी कोडॉन्स आर पॉसिबल? लेट अस ट्राय टू अंडरस्टैंड. कंसिडर सपोज here I am going to draw a little diagram Just for our understanding, I am going to show with the help of diagram to clear our concept and the possible combinations. Now consider, if each codon consists of two nitrogen bases such as adenine, 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 guanine, adenine, uracil, adenine, cytosine. Okay, in the same manner other. How many codons are possible? 4 into 4, that is 16. 4 into 4, that is 16. Right? It means that these 16 codons are also not enough to specify the 20 amino acids. It means, it proves that the codons are neither singlet nor doublet. Each codon is not made up of either a single nitrogen base or two nitrogen bases. Then comes the third possibility, that is the codon consists of group of 3 nucleotides. मग तिसरी शक्यता काय येते तर प्रत्येक कोडॉन हा 3 नायट्रोजन बेसेसने तयार झालेला असावा. सो लेट अस स्टडी अबाउट इट. लेट मी एक्सप्लेन यू. इफ codon consists of 3 nitrogen bases. How many nitrogen bases are there in RNA? 4 different types, that is 4 into 4 into 4, that is 64. Let me explain you by drawing a diagram. Here I am going to draw a table just for our correct understanding. Let us consider the nitrogen bases A, G, U, C, A, G, U, C. In this condition, we have to consider the 3 nitrogen bases in a single codon. So, what possibility is there? Let me explain you. A, G, U, C. A, G, U, C. A, G, U, C. And again here. A, G, U, C. Let us write the nature of and number of possible codons. First, A. Let me write here. A. First possible codon. Second, G. Third, AAU. Next, AAC. 4 codons. In the same manner, 4 plus 4 plus 4 plus 4, 16, and 16 4s are 64. It means that if there are 3 nitrogen bases in each codon, then 64 possible codons are there. जर त्या ठिकाणी एका कोडॉन मध्ये 3 नायट्रोजन बेसेस असतील तर एकूण 64 कोडॉन्स हे पॉसिबल आहेत. And we know these 64 possible codons are more than necessary to specify the 20 amino acid, isn't it? अर्थात हे 64 codons 20 amino acid ला स्पेसिफाय करण्यासाठी पुरेसे नाही तर त्याच्यापेक्षा पण जास्त आहे. Actually, out of these 64 possible codons, the 3 codons are called nonsense codon. 64 जे कोडॉन्स या ठिकाणी पॉसिबल आहेत त्यापैकी 3 कोडॉन्सला नॉनसेन्स कोडॉन्स म्हणतात. बिकॉज that 3 codons does not specify any amino acid, does not carry information for any amino acid. It means the 61 codons specify the 20 amino acid. याचा अर्थ उरलेले जे 61 कोडॉन्स आहेत ते 20 अमाइनो ऍसिड ला स्पेसिफाय करतात. and hence that 61 codons which specify for different amino acids are called as a sense codon, and remaining 3 which does not code for any amino acid are known as the nonsense codon. Of course, that sense codon, nonsense codon have a particular significance about which we shall talk after some time. But temporarily we have to understand why is codon triplet in nature. So here we have studied. Now we have to study about the characteristic features of genetic code, as well as before to that some important terms such as once again codon, anticodon, codogen, initiation codon, termination codon, and then the characteristics feature of genetic code. So let us talk about that important terms. Terms used in genetic code. Let us talk first term, that is codon. The sequence of 3 nucleotides or nitrogen bases in messenger RNA is known as codon. The sequence of 3 nucleotides or nitrogen bases on messenger RNA is known as codon. All of we are known that the messenger RNA, it is single-stranded and linear structure. It means that messenger RNA is made up of only single strand of ribonucleotides. It is linear, means it does not show coiling upon itself, and hence there is no question of complementary nitrogen bases. Now this messenger RNA has 5' end and 3' end, and consider these are supposed to be the sequence of nucleotides, sequence of nitrogen bases. So I said just now that the sequence of 3 nitrogen bases or nucleotides present on messenger RNA is known as codon, and each codon specifies single amino acid. Come to the second, that is initiation codon. Initiation codon. The codon present at the 5' end of messenger RNA. The codon present at the 5' end of messenger RNA is known as initiation codon, or maybe known as start codon, because this first codon present at the 5' end of messenger RNA initiates, starts the process of protein synthesis. Protein synthesis ची प्रक्रिया या 5' एंडला असलेल्या पहिल्या कोडॉन पासून सुरू होत असते, आणि म्हणून this first codon is known as the initiation codon or start codon. This initiation codon is mostly AUG, and this AUG specifies amino acid methionine in eukaryotes and N-formyl methionine in prokaryotes. N-formyl methionine in prokaryotes. Come to the next, that is termination codon. The codon present at the 3' end of mRNA. The codon present at the 3' end of mRNA is known as termination codon, because this codon, last codon, it brings about termination of process of protein synthesis. It ceases, it brings about the termination of process of protein synthesis. 3' एंडला असलेला जो कोडॉन आहे तो प्रोटीन सिंथेसिसच्या प्रक्रियेला टर्मिनेट करतो, संपवतो, म्हणून त्याला टर्मिनेशन कोडॉन असं म्हणतात. दिस टर्मिनेशन कोडॉन इज आयदर UAA or UAG. G or UGA. Out of these 3 codons, any one is present at the 3' end of messenger RNA. Let me tell you, this UAA is also known as OCA. UAG is also known as amber, and UGA is also known as opal or umber. Now, this termination codon do not specify any amino acid. They do not specify any amino acid. Means these termination codon do not bring information for any amino acid. And that's why these termination codons may be known as the stop codons or may be known as nonsense codon. So, on the other hand, we can say, except this termination codon present at the 3' end, all other codons of messenger RNA specify different amino acid. Means except this termination codon, remaining codons may be known as the sense codon because they carry information for amino acid. या ठिकाणी मेसेंजर आरएनएच्या 3' एंड वर असणारा जो टर्मिनेशन कोडॉन असतो दॅट मे बी एनी वन ऑफ दीज 3. तो कुठल्याही अमायनो ऍसिडला स्पेसिफाय करत नाही, कुठल्याही अमायनो ऍसिड साठीची माहिती आणत नाही, म्हणून त्याला नॉनसेन्स म्हणतात. आणि त्याच्या व्यतिरिक्त इतर सर्व जे कोडॉन्स आहेत मेसेंजर आरएनए वर ते कुठल्या ना कुठल्या अमायनो ऍसिडची माहिती कॅरी करतात, म्हणून त्यांना म्हटलं जातं सेन्स कोडॉन. Of course, you must know these 3 termination codons, though they are nonsense, but they have also significance in the process of protein synthesis because they terminate the process of protein synthesis. Next term, that is anticodon. Anticodon. Anticodon means the sequence of 3 nitrogen bases or nucleotides in tRNA, transfer RNA. The sequence of 3 nitrogen bases or nucleotides in tRNA known as anticodon. We people have studied about the transfer RNA in 11. Let me draw here a little diagram. Suppose here I am going to draw trifoliate model of tRNA. Just consider. The tRNA may appears in this form or tRNA may appears like this, hairpin model. Now, in this tRNA, there is a presence of sequence of 3 nitrogen bases. This sequence of 3 nitrogen bases present in tRNA, which is complementary to the codon in mRNA known as anticodon. You know, anti, this word stands for opposite. याचा अर्थ असा होतो की या ट्रान्सफर आरएनए वर असणाऱ्या या 3 न्यूक्लिओटाइड्स ला अँटीकोडॉन असं म्हणतात. आणि हे अँटीकोडॉन्स मेसेंजर आरएनए वर असलेल्या कोडॉन्सला कॉम्प्लिमेंटरी असतात. म्हणजे समजा इफ ऑन मेसेंजर आरएनए द कोडॉन इज AUG. So the anticodon of this AUG codon is of course complementary to it, that is UAC, because we know that the complementary nitrogen bases are A, U, G, C, isn't it? So these anticodon play very important role at the time of protein synthesis, about which we shall discuss in next topic, that is translation. One more term, last term, codogen. Codogen, it is the smallest possible sequence of nucleotides present on DNA strand and specifies one amino acid. Going to repeat. Codogen means it is smallest possible sequence of nucleotides present on DNA strand and specifies one amino acid. Listen, suppose here is a double-stranded DNA. Consider this is first nucleotide, second nucleotide, third nucleotide. Now this sequence of three nucleotides present on strand of DNA which possess information for single amino acid is known as codogen. This codogen may be known as NODOC. Now one more thing, that the codogen and codons are complementary to each other. याचा अर्थ कोडोजन आणि कोडॉन हे परस्परांना कॉम्प्लिमेंटरी असतात. कसं? लेट अस सी. सपोज on this DNA strand here is a presence of GGG nucleotide sequence, nitrogen based sequence, and whatever mRNA is synthesized on it, आणि या स्ट्रँडवर जो मेसेंजर आरएनए तयार होईल, अर्थातच त्या मेसेंजर आरएनए वर जीजीजी याच्या कॉम्प्लिमेंटरी नायट्रोजन बेसेस येतील. म्हणजे या ठिकाणी येणार आहे CCC. Isn't it? And we know the triplet of nucleotides on messenger RNA is called as the codon. That's why I said that the codogen and codon are complementary, and codon and anticodon are also complementary. So here we have first of all discussed only about the important terms used in genetic code. Now let us talk about the characteristics of genetic code. Characteristics of genetic code. First, triplet nature. It means genetic code or codon is triplet in nature. That is, the codon is formed of group of three successive nucleotides, or codon represent the sequence of three nitrogen bases. That is triplet nature, and we have already studied. Come to the second. Genetic code is commaless. Genetic code is commaless. Let me explain you by drawing the diagram. Consider as usual, here is a messenger RNA with sequence of nucleotides 5' end, 3' end, and now let us talk about the second characteristic. First characteristic, I have already told you that the codon or genetic code always show triplet nature. It means the codon is formed of sequence of three successive nitrogen bases. So here is the first codon, second codon, third codon, in this manner. Commaless. The codons or genetic code is commaless, means there is no comma, there is no pause, there is no gap, there is no punctuation, there is no space between two successive codons. एका पाठोपाठ एक येणाऱ्या कोडॉन्सच्या मध्ये कुठे कॉमा नाहीये, कुठे स्पेस नाहीये, गॅप नाहीये. याचा अर्थ काय? All the codons are continuous from 5' end to the 3' end. This characteristic is called commaless. Genetic code is commaless. Next, non-ambiguity or non-ambiguous nature. Non-ambiguity or non-ambiguous nature means what? A particular codon specifies particular amino acid only. Particular codon specifies particular amino acid only. याचा अर्थ विशिष्ट कोडॉन विशिष्टच अमाइनो ऍसिड ला स्पेसिफाय करतो. Particular codon, it carries information for specific amino acid. It means one codon do not carry the information for two different amino acid. याचा अर्थ एक कोडॉन दोन किंवा दोनापेक्षा अधिक अमायनो ऍसिडला स्पेसिफाय करणार नाही. दोन किंवा दोनापेक्षा अमायनो ऍसिड साठी इन्फॉर्मेशन कॅरी करणार नाही. Each codon is specific for particular amino acid. Say, for example, the GGG is the codon. Consider GGG हा एक codon आहे, and this GGG codon always specifies the amino acid glycine. हा नेहमी ग्लायसिन या अमायनो ऍसिडला स्पेसिफाय करेल. It means GGG, this codon will not carry the information for any other amino acid. It means it is non-ambiguous. Non-ambiguous याचा अर्थ होतो की निसंदिग्धता. त्यांच्यामध्ये संदिग्धता दिसत नाही, डुप्लिकेट नेचर दिसत नाही, हे कृपया लक्षात घ्या. बट देयर इज वन एक्सेप्शन. एक एक्सेप्शन आहे, तेवढं मात्र मी सांगतो. Few minutes before I told you, the initiation codon is always AUG, and this AUG, it carries the information for methionine. It specifies for amino acid methionine. And GUG, it is another codon. It specifies amino acid valine. But sometimes if AUG is not available as an initiation codon, पण जर काही वेळेला एक्सेप्शनल केसेस मध्ये AUG हा इनिशिएशन कोडॉन म्हणून जर अवेलेबल नसेल, तर त्याच्या जागेवर GUG हा इनिशिएशन कोडॉन म्हणून काम करतो. and though it is GUG but it specifies the amino acid methionine. म्हणजे एवढंच फक्त एक एक्सेप्शन आहे की ज्यामध्ये GUG हा कोडॉन व्हॅलिन बरोबरच आवश्यकता असेल त्यावेळेला मिथ्योनिनला सुद्धा स्पेसिफाय करतो. इट ओनली शोज द ambiguity. Otherwise all other codons are non-ambiguous. This is called non-ambiguous nature. Next, that is polarity. The genetic code shows polarity. That is, the genetic code or codons are unidirectional. It means the codons can be read only in one direction, that is from 5'N to the 3'N. Genetic code or codons are unidirectional, or they show polarity. It means they can be read only in one direction, always in one direction, and that is from 5'N to the 3'N. This is called polarity. याचा अर्थ असा होतो की जेनेटिक कोड किंवा कोडॉन्स हे पोलॅरिटी दाखवतात. ज्या वेळेला ट्रान्सलेशनची प्रक्रिया होते, प्रोटीन सिंथेसिस सुरू होतं, त्यावेळेला रायबोझोम हा नेहमी या मेसेंजर आरएनए वर असणाऱ्या कोडॉन्सला 5'N पासून रीड करायला सुरुवात करतो, and it is carried out towards the 3'N. That is in unidirectional. That is unidirectional way. This is called as the polarity. Next, degeneracy. Genetic code are degenerate. It means the information for one amino acid may be carried by more than one codon. Please don't get confused. Here in this characteristic we have seen one codon specifies one amino acid. या तिसऱ्या कॅरेक्टरिस्टिक मध्ये आपण असं शिकलो की एक कोडॉन एकाच अमायनो ऍसिडला स्पेसिफाय करतो, पण या ठिकाणी आता आपण म्हणतोय की वन अमायनो ऍसिड कॅन बी स्पेसिफाइड बाय मोर दॅन वन कोडॉन. व्हाय? बिकॉज वी नो दॅट in all 64 possible codons are there. We have studied in previous topic. एकूण 64 codons हे possible आहेत, but out of that 64 possible codons, 3 codons are nonsense. Means 3 codons does not specify any amino acid. So remaining 61 codon, they carry information for 20 amino acid. याचा अर्थ 61 कोडॉन्स अवेलेबल आहेत, 61 कोडॉन्स पॉसिबल आहेत आणि त्यांना फक्त 20च अमायनो ऍसिडला स्पेसिफाय करायचंय, 20 अमायनो ऍसिडला फक्त स्पेसिफाय करायचंय. सो ऑब्वियसली one amino acid can be specified by more than one codon. याचा अर्थ एका अमायनो ऍसिड ची माहिती एकापेक्षा जास्त कोडॉन कॅरी करू शकतात. This is called as the degeneracy. Let me explain you. Previously I told you GGG. This codon specifies amino acid glycine. ठीक है? But in addition to that, another codon GGC, GGA, GGU, they also specify the same amino acid. Okay? It means one amino acid can be coded by more than one codon. This is called as the degeneracy. याला degeneracy असं म्हणतात. एक उदाहरण सांगतो. Exceptional. Amino acid methionine is coded by only single codon AUG, while amino acid tryptophan is coded by codon UGG. These are exceptional cases. म्हणजे एकूण 20 अमायनो ऍसिड पैकी हे असे 2 अमायनो ऍसिड्स आहेत की ज्यांना स्पेसिफाय करणारे एकच कोडॉन आहेत. बाकी 18 अमायनो ऍसिड्स can be coded by more than one codon, and this characteristic is known as the degeneracy. Genetic code is degenerate. Move to the next character. Genetic code is universal. It means the given codon specifies particular amino acid in all living organism. Given codon specifies particular amino acid only in all living organism, that is from simple prokaryotic bacteria up to the highly advanced human being. अतिशय साध्या सजीवांपासून बॅक्टेरियांपासून ते मानवापर्यंतच्या सर्व सजीवांमध्ये विशिष्ट कोडॉन हा विशिष्टच अमायनो ऍसिडला स्पेसिफाय करत असतो. देयर इज नो चेंज एक्सेप्ट इन ईस्ट अँड इन सर्टन मायकोप्लाज्मा देयर आर सर्टन एक्सेप्शन्स. अदरवाइज इट इज युनिव्हर्सल, मीन्स the genetic code is applicable to all living organism except yeast and certain mycoplasmas. Come to the next characteristic, that is Obel phenomenon. Obel phenomenon or Obel hypothesis. This hypothesis was proposed by scientist Francis Crick in year 1966. Few minutes before we have seen that the anticodon present in tRNA is always complementary to the codon in mRNA. tRNA वर असलेला जो anticodon आहे तो mRNA वर असलेल्या कोडॉनला कॉम्प्लिमेंटरी असतो. This is because of A, U and G C nitrogen base pairing. Now the ability of transfer RNA, listen, the ability of transfer RNA to form pairing to the more than one codon in messenger RNA due to unusual base pairing present at the third position is known as Obel phenomenon or Obel hypothesis. परत एकदा मी रिपीट करतो. I said that the ability of transfer RNA to form pairing with more than 2 different types of codons in messenger RNA is due to the unusual base pairing at the third position. Now listen. Suppose consider here is a Anticodon on messenger RNA. कल्पना करा या ठिकाणी मी मेसेंजर आरएनए वरचा कुठला पण एक कोडॉन लिहितोय. आपण जी 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 हा कोडॉन कन्सिडर करू. So as we know that the anticodon in tRNA is complementary to the codon. So what would be the anticodon for this codon? Obviously the anticodon present in tRNA will be CCC. Okay, now here you can see that there is a precise matching, there is a complementary matching between the nitrogen bases of codon and nitrogen bases of anticodon, but this anticodon may show pairing with certain other codons, such as here I am going to write few other codons: G G A, G G C, G G U. Here I have written three different types of codons. Now the same anticodon CCC can form the pairing with these three different types of codons. Now here in this pairing of anticodon and codon, the complementary base pairing of first two nitrogen bases is precise, is exact. Okay, while there is no precise complementary base pairing present at the third position. आता या ठिकाणी आपल्या लक्षात आलं असेल की ज्या वेळेला हा अँटीकोडॉन या अशा प्रकारच्या तीन कोडॉन्स बरोबर पेरिंग करतो त्यावेळेला पहिल्या दोन पोझिशनला असलेल्या नायट्रोजन बेसेस मध्ये प्रिसाइज एक्झॅक्ट पेरिंग आपल्याला पाहायला मिळतं. अँड देयर इज नो प्रिसाइज, नो एक्झॅक्ट, नो कॉम्प्लिमेंटरी बेस पेरिंग प्रेझेंट ॲट द थर्ड पोझिशन. अँड देअरफॉर this nitrogen base present at the third position is called as the obal position, obal base, and phenomenon is known as the obal phenomenon or obal hypothesis. You know the meaning of obal. Obal याचा अर्थ होतो not fixed, changeable, बदल होणारा. So nitrogen base present at the third position in codon may not be fixed, but then and then the same anticodon can form the pairing with such different types of codons. This phenomenon is called as the obal phenomenon. So here we have understood the genetic code and all the characteristic features. Let us stop here. Thank you.